All right, so it's 9 o'clock. Hello, everyone. Welcome to our today's museum seminar. So before I get started, I just want to make a short announcement. So since this week, we actually entered the summer period, and then we're gonna resume our twice a week summer museum program. So hopefully I'm gonna see you all guys again. So one time is still the regular time during the week, and the one is during the weekend. Okay, so it's absolutely my pleasure to welcome our today's speaker, Dr. Xinzhong Dong from Department of Neuroscience, Johns Hopkins University. Well, I think Xinzhong is one of my favorite scientists in the field. I mean, it's his work is absolutely elegant. Uh, Not only the previous neuroscience work, but also currently he's now switched to a little bit to the immunology field, which I think the work also quite marvelous. Xinzhong completed his PhD training in UCLA, in the cell cycle and the neuroscience, and then following with the postdoc training in neuroscience in Caltech. And then from 2011, Xinzhong started his independent research program as assistant professor in Department of Neuroscience in Johns Hopkins. And then all the way up to 2000, sorry, Xinzhong actually started his research program from 2004, and then all the way up to 2015 as a full professor in Department of Neuroscience and Dermatology, Neurosurgery in Johns Hopkins. And then, meanwhile, he was also elected as early career scientist in Howard Hughes from 2009, and then from 2015, he was also elected as investigator in Howard Hughes Medical Institute. So I think a lot of us probably already quite familiar with Xinzhong's work. His lab is taking really multidisciplinary approach to understand the cellular and the molecular machinery of pain and itch, which are initiated and then mediated by the primary sensory neurons in dorsal root ganglia. Xinzhong's lab actually identified a novel family of G protein coupled receptors in mouse called MRGPRS. I always find this name a little bit hard to pronounce, but now it's actually proved it's quite really important both in neuroscience and immunology. So his lab actually found several MRGPRS are specifically expressing the DRG neurons and function as novel itch receptors. But as I mentioned, from the beginning to today, Xinzhong's lab actually start to appreciate that these receptors not only function significantly in the neurons for the itch sensation, but now its research focus on these receptors also switched to the immune cells, such as the mast cells. These receptors also contribute significantly for these innate cell activation and for the whole systemic whole body immunity. So the other major contribution from Xinzhong's lab, including but not limited, identification of itch receptors, as I mentioned, like the MRGPRS family, and also identification of itch sensing neurons. And also, his lab also identify these the regulators for TRP channels called PIRT and PRT2. And then he developed a functional imaging of DRG neurons and study persistent pain. And the last but not least, his lab also studying mast cell and neutrophil specific receptors. So with these really groundbreaking discoveries, Xinzhong was awarded as numerous honors, which I'm not gonna list all of them. But I just give a few examples. He was awarded as a Darling Long Pain Service Award, Jarvis Neuroscience Investigator Award from NIH, and then just last year it was elected as a AAAS Fellow. I think one more things I want to mention here is Xinzhong has extremely successful training record for the postdoc and PhD students. I think so far, I think dozens of his postdoc fellows move on and take independent faculty positions, which I found is extremely impressive. So I think without further ado, I think Xinzhong gonna talk about the GPCR family Mrg receptors today with us to introduce or the function in, I think, both neuroscience and immunology. So thank you so much, Xinzhong. We're looking forward to talk.
Oh, thank you very much, Chuan, for the generous introduction. So let's try introduce. So my lab focus on work this novel GPCR family called Mrgs in pain, itch, and innate immunity. And sorry, advance. Yeah, so the Mrgs encode a large family of GPCRs, and shown here are the mouse and human Mrgs, and there are 50 mouse Mrgs and 18 human Mrgs. And so Mrg genes are actually clustered on mouse and human chromosomes. And so each bar shown here represent one Mrg genes. So the illustration also summarizes the expression pattern and the ligand for the Mrgs. My lab has deorphanized for the past 20 years. So the Mrgs initially identified in sensory neurons, and more recently, some of them are expressed in immune cells. So they play an important role in diverse biology and diseases, as I will show you later. So based on the ligand specificity and the expression, we can identify the human homolog shown here. So let me talk about the role of Mrg in as an itch receptor first. So what is itch? Itch is also known as a pruritus, and is defined by this German physician several hundred years ago, and he defined itch as an unpleasant sensation to elicit the desire to reflex to scratch. So it's pretty accurate and simple definition we still use today. And in general, we believe itch provides us a protective mechanism. For example, if you're scratching the skin, you can remove the irritant from the skin. And people also believe the scratching induced mechanical pain or sensation can also mobilize immune cells in the skin. So there are among all the somatosensations such as pain and touch, itch is the least understood, and it is unclear whether itch is a distinct sensation at the cellular molecular level. And although histamine is the best characterized itch mediator, the majority of chronic itch conditions are non-histanergics. So it is also unknown whether there's a itch receptors mediating non-histanergic itch. So the work from my lab and many other lab has demonstrated itch. It is a distinct sensation from pain, and they mediate by itch specific neuron in dorsal root ganglion or DRG shown here. And for example, my lab found some of the MRG shown here expressed specifically in sensory nerves innervating the skins, and they function as a novel itch receptor by sensing different itchy substance in the skin, including peptides, amino acid, and medicines. So identification of these receptors really open a new avenue to study histamine-independent itch, which again represent the majority of chronic itch. So many diseases can cause chronic itch, and shown here, there's a list of different diseases cause itch. So you're probably familiar with the skin conditions like eczema, psoriasis, different type of dermatitis. In addition, systemic disease can also cause itch. So for example, liver disease patient and kidney disease patient often suffered from chronic itch. So today I'm going to talk about liver disease cause itch. In addition, drug side effect, itch is one of the major drug side effects. For example, morphine can cause a lot of itch, and many other drugs can also cause itch as a side effect. A nerve injury, psychogenic itch is also a major contribution for chronic itch. So over 2,000 years ago, a Greek physician made a striking observation. So people whose skin turn yellow commonly complained about severe itch. So today, we know the yellow skin is a condition called the jaundice, and it's caused by the buildup of bilirubin. So bilirubin is a yellow chemical degraded from heme and can be excreted in the urine and feces. And however, when the liver is under a disease condition, bilirubin excretion is blocked. So the bilirubin level in the body can be at the micromolar range and cause jaundice. And the jaundice-associated itch commonly occur in the context of cholestasis. So cholestasis is a condition of slow stoppage of the bile flows, and it can cause by different liver conditions. And many people suffered from this condition. However, there's no effective treatment for cholestatic itch, and the underlying mechanisms unclear. So a former MD PhD student, Jimmy Meixiong, asked a simple question: does bilirubin cause itch or not? So we can use mouse as our model system to study itch. So how do we really study itch? In mice, it's quite simple. So I apologize, the video doesn't play. But basically, we inject a small amount of itchy substance into the skin. If the mice feel itch, they will use their hind paw to scratch the injected site at a high frequency. So for example, you can inject histamine or chloroquine. In this case, we inject bilirubin in the skin, and then we videotape the mouse for like 30 minutes, so you can count afterward the number of scratching they elicited. Oh, so actually play it, and so you can see they scratched. Yeah, it's pretty robust and characteristic behavior. So Jimmy did the experiment, so inject again with a bilirubin into the skin. As you can see here, bilirubin injection in the wild type mice really induce a dose-dependent scratching. As the dose increase, the scratching increase. More importantly, if you treat the mice with antihistamine, it doesn't inhibit bilirubin-induced itch. This suggests bilirubin causes a non-histaminergic itch. And then Jimmy asked whether there are any bilirubin receptor exist. So he screened the bilirubin against all MRGs, and he found bilirubin can specifically bind and activate mouse MRGA1. In human, it's called the MRGX4. Here, using a calcium imaging assay, and then we also tried the ligand binding assay. So the EC50 and the KD value are in the micromolar range, which are consistent with the level of bilirubin in jaundice patient. So as a common feature, most MRGs are low affinity and broadly tuned receptor to monitor high level of damaging internal or external substances. And so this is the first bilirubin receptor ever discovered. So Jimmy also found MRGA1, the mouse receptor, is specifically expressed in a subset of DRG neurons shown as a green staining here. So then we did electrophysiologic recording to look at the action potential of DRG neuron activation. As you can see here, bilirubin can induce a robust action potential in wild-type DRG neuron. And we also generated MRGA1 single gene knockout mice, and the bilirubin failed to induce any action potential in MRGA1 knockout DRG neurons. And more importantly, When he inject the skin with a bilirubin again in the knockout MRGA1 knockout animal, the bilirubin-induced scratching is significantly reduced in the MRGA1 knockout mice, suggesting the MRGA1 mediating bilirubin-induced itch. And Jimmy also employed a mouse model of cholestasis by treating the animal with a drug to damage the liver. So as you can see here, under this model, there are significant increase of bilirubin buildup in both wild type and MRG knockout mice as compared to the healthy controls. And interestingly, the wild type mice with a cholestatic condition scratch significantly more than the healthy mice. And however, the MRGA1 knockout mice with a cholestasis scratch significantly less than the wild type. So this data indicate the MRGA1 mediate cholestatic itch. And the skin bilirubin level, we also find actually correlate significantly with the level of scratching. On the other hand, if we measure the serum bilirubin level, we see a trend, but no significant correlation. We also collaborate with a physician scientists who provide us serum from 6 healthy individuals and serum from 6 patients suffered with cholestatic itch. Some of the parameter of those patients shown here. As you can see, the bilirubin level is really high in those patients, and they also experience a strong itch. And then Jimmy actually injected these serums from either healthy controls or the patient suffered with cholestatic itch into the either wild type or MRGA1 knockout mice. So the healthy control serum did not induce a significant increase of itch. On the other hand, the serum from the cholestatic itch patient induced a very strong scratching in the wild type mice, and that scratching are significantly reduced in the MRGA1 knockout mice. As maybe you may notice, the knockout still have some significant scratching, so suggesting there's probably other mechanism or other mediator contributing to cholestatic itch. And Jimmy also took multiple approaches to deplete the bilirubin from the patient serum to show the bilirubin in the patient serum really causes itch from MRGA1. So he used 2 methods. One is using iron chloride to deplete bilirubin or anti-bilirubin antibody to deplete bilirubin. So in both cases, there's a significant reduction of scratching after the depletion of bilirubin. So here, just summarize this part of the talk. And so we found that both human and mouse MRGs are bilirubin receptors. Later on, in a separate study by Jimmy and also Li Yulong's lab in China, we also find the human MRGX1 is a bile acid receptor. So this receptor not only can be activated by bilirubin, it also can be activated by different bile acid. So we think this receptor mediate cholestatic itch in both mice and human. So beside this itch receptor in sensory neuron, we found that recently some of MRGs are specifically expressing immune cells, such as mast cell, neutrophil, or basophil. So we identify at least 2 MRGs specifically expressing the innate immune cells. One is called MRGB2 in mice, specifically expressing mast cells. Other one is called MRGA2, expressing neutrophil. So today I will show you several published and unpublished study that contribute to itch, inflammation, and pain, and host defense against bacterial infections. So let's talk about the mast cell project first. So mast cells are the tissue resident cells, and they are the first responding cells upon infection or injuries. So when mast cells are activated, they release and degranulate and release many pro-inflammatory mediator like histamine, serotonin, proteases, cytokines, and this will result in recruitment of many other immune cells like a neutrophil, and to induce inflammation. So there are two main pathways to activate mast cells. One is the antibody IgE and the receptor Fcs to induce a classic allergic reactions. On the other hand, IgE independent pathway is also well documented. People find numerous positive charged peptides and substance compounds, which collectively called the basic secretagogues, can also activate mast cells. So despite of decades of search, the receptor for basic secretagogues was unknown until our discovery. So a former postdoc, Benjamin McNeil, identified a mast cell specific receptor, and he used RT-PCR to detect only the mouse MRGP2 is specifically expressed in mast cells. So if you look at here, we find only MRGP2 is expressed in mast cells. And then he also generated MRGP2 Cree mice using a Cree TD tomato labeling. He found MRGP2 shown as a red staining, nearly 100% expressed in mast cells, co-labeled with avidin. So MRGP2 Cree mice is a great tool to study mast cell biology. And this is consistent with online gene chip data shown here. So mouse MRGP2 is exclusively expressed in mast cell, but not in DRGs, not in any other tissue cell types. So it's very specific mast cell specific receptor. On the human side, people and our lab found MRGX2 is specifically expressed in human mast cells. So importantly, Ben used multiple approaches to demonstrate human MRGX2 and mouse MRGP2 is a receptor for basic secretagogue shown here on the list. And here just show the EC50 of some of the basic secretagogue, including the famous compound called the compound 4080, neuropeptide substance P, which I'm going to talk about later, and several other peptides, positive charged peptides. In the same study, Ben also find many drugs shown here with anaphylactic reaction side effect can also activate these two receptors at the EC50 within the clinical doses. So what is anaphylactic reactions? So anaphylactic reaction can affect many people in the US with life-threatening symptoms, such as drop in body temperature, low blood pressure, and the shortness of breath. And it can cause by many medications, food, and venoms. And strikingly, Ben found many of these drugs with anaphylactic reactions can induce a robust activation of wild-type mast cells shown here. So nearly 100% activation of mast cells. And the activation is completely abolished in the MRGP2 knockout mast cells. And Ben also showed the drug induced anaphylactic reactions indicated here by the drop of body temperature are completely gone. So in this study, he used a drug called the ciprofloxacin. This drug, if you go IV injection at a higher level, it will induce very strong anaphylactic reactions in both human and here in mice. And many of these wild-type mice actually died from the reaction. However, in the MRGP2 knockout mice, the anaphylactic reaction is completely abolished. So suggesting MRGP2 mediate drug-induced anaphylactic reactions. Two recent study, one from Brian Ross' lab and another from Sun Jinpeng's lab, and they actually solved the 3D structure of MRGX2 and MRGX4, the each receptor I mentioned previously. So here the structure for X2. So by cryo-EM, this finding actually confirmed our discovery. So they showed the MRGX2 has a very shallow binding pocket, which is consistent with a notion that this receptor are promiscuous receptor and combines to the shallow binding pocket combines to many ligands. In addition, a ligand has a negative charge residue in the binding pocket for interaction with a positive charge ligands. So it is well documented mast cell play an important role in pain and itch. A former postdoc, Dustin Green, investigated contribution of MRGP2 and MRGX2 in mast cell to pain sensation. So upon tissue damage, a sensory nerve can release a neuropeptide called the substance P, and to activate nearby mast cells to induce a neurogenic inflammation and pain. So the reason they call neurogenic inflammation is the source of the inflammation actually comes from the nerves. So that's why they call the nerve neurogenic inflammation. However, it is unclear what receptor on the mast cell mediate this process. So the dogma in the pain field suggests the substance P canonical receptor is called NK1 receptor or tachykinin receptor, is a receptor mediate substance P induce neurogenic inflammation and pain. However, the outcome of the clinical trials using NK1 antagonist is very disappointing and without obvious analgesic effect. So basically, the antagonist targeting this canonical substance P receptor did not block pain. So what is going on here? To address this discrepancy, Dustin did a simple experiment by RT-PCR, using RT-PCR and find a surprising result. So the NK1, the canonical receptor, is only expressed in spinal cord, but not in the DRG, not in the mast cells. On the other hand, our MRGP2 receptor is highly expressed in mast cells, but not in DRG and the spinal cord. And consistently, when he gave a substance P, the peptides, onto either wild type or MRGP2 knockout mast cell, you can see quite a difference. So the substance P can induce a robust mast cell activation in wild type mast cell, but the reaction is completely gone in the MRGP2 knockout mast cell. So MRGP2 is essential for substance P induced mast cell activation. In addition, substance P can induce a mast cell release of cytokines such as the CCL2 and CCL3 seen in the wild type mice, and this CCL2, CCL3 release is completely gone in the MRGP2 knockout mice. And the skin injection of a substance P can also recruit immune cells, and so Dustin actually inject the substance P into the wild type or NK1 knockout or MRGP2 knockout mice. In both wild type and NK1 knockout mice, substance P injection can induce robust immune cells recruitment, and that recruitment phenotype is completely abolished in the MRGP2 knockout mice. So besides the direct injecting the substance P into the skin, Dustin also employed the tissue injury model by making a small incision into the mouse hindpaw. As shown here, you can see that the tissue injury-induced inflammation is pretty robust in the wild-type mice, whereas in the MRGP2 knockout mice, the injury-induced inflammation is significantly reduced. In addition, the injury-induced mechanical hyperalgesia or mechanical pain is also significantly reduced compared to the wild-type mice. So indicating the MRGP2 contribute to inflammation and pain. And consistently, injury-induced immune cell recruitment. So here is a hindpaw incision model. So in the wild-type mice, and there's a lot of immune cell recruitment. So that's why you see the inflammation in the hindpaw. The knockout hindpaw is have a significant reduction of tissue immune cells recruitment after injury. So here, just summarize this part of the study. So we think after nerve sensory tissue injury, sensory nerve actually secrete a substance P, which can activate nearby mast cell through this mast cell receptor MRGP2 to release cytokine chemokine to attract or recruit more immune cells into the injury site to induce neurogenic inflammation and pain sensitivity. So in a separate study, Jimmy, the former student, also compared the contribution of the MRGP2 and IgE, the canonical mast cell activation pathways, to each sensation. So because of time, I just summarized his finding. So he found these two pathways cause the mast cell to release different content. On one hand, the IgE pathway release more histamine and serotonin from the mast cell. On the other hand, the MRGP2 pathway release more tryptase and less histamine and serotonin compared to the IgE FC pathways. Interestingly, these two pathways activate different sensory nerves nearby. So this data demonstrate that MRGP2 in the mast cells can mediate non-histaminergic itch. So now I want to switch gear to talk about another immune cell type MRG expressing is a neutrophil. So this is a defensin receptor in neutrophil, which can prevent dysbiosis in the skin and bacterial infection. And this study is done by a former postdoc, Xinzhong Dong, now is a faculty at UT Dallas. And as many of you know, defensin are antimicrobial peptides. They were first identified from frog skin in 1987, and defensin has a very unique property. They can directly kill bacteria and fungi. And there are more than 50 mouse defensins and human defensins in their genomes. And they express in the mouse human skins and also GI tracts, and so where they can defend against the infections. However, due to the gene redundancy among the defensin genes and lack of genetic tools, so the in vivo mechanism of defensin action is unclear. So we don't know whether defensin can also activate our host cells, and if so, it's unknown whether the host cell has a receptor for defensin. So the defensin genes, like MRGs, also clustered a small region on mouse chromosome. Again, there's 50 of individual defensin genes, span about 3 million base pairs on the mouse genome. To really solve the gene redundancy problem, and Xinzhong generate a conditional knockout line by insert a LOXP site at each end of the entire defensin gene cluster, again, about 3 million base pairs. And then she used a keratinocyte-specific CRE called the K14 CRE to delete all 50 defensin genes. So it took us 2 years to make this mouse line, and pretty amazing. It worked, and RT-PCR data suggests all the defensin genes actually deleted from the skins. And so she also did gene mRNA expression analysis and find after this after the Staph aureus infection, there's upregulation of defensins, and so there's different defensins. We look at it, and defensin 3, beta 3, beta 4, beta 6, and beta 14. However, in the defensin cluster knockout mice, the expression of defensins are completely abolished. So here is a defensin peptide staining using anti-defensin antibody. And so you can see after Staph aureus infection of the skin, there's a pretty dramatic upregulation of defensin compared to the wild type skins. And the defensin cluster knockout skin is lack of any defensin stainings. To make a long story short, Xinzhong also identified the MRGA2 is a defensin receptors. So by RNA scope, and she found MRGA2 expressed in nearly 40% of all neutrophils and co-labeled with Ly6G, and the MRGA2 knockout mice lack of the gene expression in the neutrophils. And more importantly, the defensin can induce a dose-dependent neutrophil degranulation assayed by the release of elastase, and that release is completely abolished in the MRGA2 knockout neutrophil. So healthy skin can maintain a balanced and a diverse microbiomes, which is important to fight against infections. However, a pathological disturbance can lead to outgrowth of pathogenic bacteria at the expense of other diverse commensals. So here we perform the 16S RNA ribosomal RNA sequencing of bacteria in the naive skins. So as you can see here, the wild type mice has a significant higher number of bacterial species, and then the defensin cluster knockout skin and also the MRGA2 knockout skins. So more importantly, the proportion of the pathogenic bacteria, such as the Staph aureus, represent as this green bar, is expanded in the defensin knockout MRGA2 knockout skins as compared to the wild type skins. So this data demonstrate both defensin and its receptor MRGA1 maintain a skin microbiome diversities and prevent skin dysbiosis. So beside the naive skin, Xinzhong also examined the skin after Staph aureus infections. So she found both defensin knockout MRGA2 knockout mice exhibit a pretty similar phenotype. So the mutant mice have a more severe bacterial infection shown as the lesion size. So the representative image shown here. So the knockout has a significantly bigger lesion size compared to the wild type mice. In addition, the bacterial load after the infection is also significantly higher in the two knockout mice compared to the wild type mice. Here are the representative the image. So while the knockout has a much higher bacterial load after infection. So in addition, Xinzhong did skin histology after Staph aureus infections. So the hallmark of Staph aureus immunity is a rapid formation of this neutrophil abscess. And so grand staining shown here in the wild type skin 24 hours after Staph aureus infection. So the bacteria actually confined in the abscess, surrounded by many neutrophils stained by this Ly6G staining. However, in both defensin knockout and MRGA2 knockout mice, the bacteria actually spread to a very large area in the skin due to the lack of the abscess formation by neutrophil. So here is a quantification of a neutrophil recruitment. So wild-type mice, after infection, there's a high level of neutrophil recruitment. In the knockout mice, the Staph aureus infection induces a significantly less neutrophil recruitment. Mechanistically, we found that the defensing can induce the release of pro-inflammatory cytokine like IL-1 beta, CXCL1, and the CXCL2 release from neutrophil in the wild type skins, and the knockout skin, the release of this mediator are significantly reduced. So suggesting the defensing and MRGA1 mediate cytokine chemokine release. So together, this data with many other data, which I don't have time to show you, we proposed the following models for the action of defensing and the MRGA2. So under healthy skin conditions, the low level of defensins and neutrophil maintain this microbiome homeostasis in the skin. However, after infection, defensins upregulated and activate the receptor MRGA2 in neutrophil to cause the release of cytokine, chemokine release, and recruit more neutrophil to form the abscess and to contain the bacterial infections. So here, just summarize the two immune cells project. So the MRGB2 in mast cell play an important role in neurogenic inflammation and the pain. So next, I'm gonna show you unpublished data on the stroke. And on the other hand, MRGA2 in neutrophil play an important role in the bacterial infections and other skin condition like psoriasis. So more recently, a current MD PhD student, Ruchita, and the neurosurgeon Zhisheng in our lab and studied the role of MRGB2 in post-stroke inflammation. And the previous study has identified mast cell in the brain meninges and as the mediator of inflammation after stroke. And consistently, we found MRGB2 shown as a MRGB2 TD tomato mice are present in the meninges and co-stained with toluidine blue. And we also did flow cytometry and found dura or meninges has many MRGB2 positive mast cells. So the peritoneal cavity mast cells serve as a positive control. To our surprise, the brain itself doesn't contain any mast cells. So next, we employed a mouse model of a ischemic model, stroke model. So 2 days after the stroke, and we performed this TTC staining to visualize the stroke-affected tissue shown as a white stain. As you can see here, the MRGB2 knockout mice exhibit a attenuated stroke volume compared to the wild-type mice. And here is a quantification after quantification of stroke volume. So the MRGB2 knockout mice has a significant reduction of stroke. And in addition, we also look at the neutrophil recruitment into the stroke area. So the MRGB2 knockout mice has a significantly less neutrophil recruited in the stroke area. Interestingly, substance P, the ligand of MRGB2 I mentioned earlier, is also upregulated in the brain after stroke. And so, which suggesting they may play a role by activating MRGB2 in mast cells in the meninges. So we have been collaborating with several companies and NIH to develop small molecule drugs targeting against different MRGs, including the MRGX4 antagonist for treating cholestatic itch I mentioned earlier, and the antagonist for MRGX2 to treat inflammatory conditions. So both of these antagonists are in the phase 2 clinical trials now. Hopefully we can get some results soon. And so the MRGA X1 I didn't mention, we also develop an allosteric modulator to treat chronic pain. So here, just summarize all the study I mentioned. So we found MRGs are expressed in sensory neuron and also in some of innate immune cells like a mast cell, neutrophils. And so they play a quite diverse roles in biology and the disease. So one of the common theme of this receptor is defense, I think. So either in sensory neuron function as an itch receptor to defend the irritant infections. In the immune cells, like mast cells, basophils, and they actually respond to infections. So yeah, so that's all the data I'm gonna show you, and I would like to thank all the people who have done the work and our collaborators and also funding agencies. Thank you very much for your attention.